What's your major?
I think we're sharing everything. So
Which note?
good.
Yeah, if you look, you can see that what it's showing.
Biochem major. Oh, I just finished it.
It started.
Yep.
Yes. We're probably. Okay, well, thank you for all your patience while I'm trying to figure out things with the computer and, like troubleshooting as well, the things you have to do, learn in research, and I just show you how I can troubleshoot, finding the right resource. Okay, so, well, if you missed the party, that was the party just took place last Wednesday, and so welcome to the Prize Lab, aka the best lab ever. And I'm saying that because I know nobody will hate me because we're all our own people here. Okay, so yeah, so you guys are going to hear me a lot today. So in case you get sick of me, I have something like somebody else to start talking. But first off, let's welcome the new undergrad-ish, but not new, because there's half new here and completely new. So for all those who haven't gone through the entire training, let's just introduce yourselves a little bit before we got into the project. Okay, so start from Noah.
Okay, I don't know what to say. I'm Noah. I don't know. I just finished technically my freshman year, but credits wise, it's weird. So I'm technically a sophomore, but who cares? I like to sing. I like to act. I like to cook. If you ever want to cook, let me know.
Well, guys, you can ever always bring us whatever you're experiencing to the lab, and we will love to be your guinea pig. Cool, Benjamin.
Yeah, my name is Ben. I'm from Sandy, Utah. I just finished my sophomore year, and I love doing anything outside, climbing, rappelling, sometimes running, depending on the day.
Ooh, running. We have a lot of running in the lab. A lot of running. I am not the one, as you can tell. Cool. Oh, sorry. Did you mention your major?
Biochemistry.
And Noah?
Biochemistry.
Okay, the best major.
I'm Spencer. I'm a neuroscience major, sadly.
You can cover yourself later.
I'm a senior, and I'm the exact opposite. I'm really indoorsy. So yeah, watching TV, video games, reading, stuff like that.
Yeah, okay, I'm the same.
I'm Hannah Morgan. I'm probably changing to biochem as of next semester. I'm a sophomore, and I'm also into the whole rappelling, rock climbing, crazy outdoorsy person.
I'm Tyler. I'm a biochem major. I'm a sophomore, I suppose, because I just finished my junior year. I just finished my sophomore year. And I like to read and swim and play quite a bit. So.
Yeah, I'm Alex. I'm a biochemistry major. I just finished my freshman year. Yeah, I'm not really too into the outdoor stuff, honestly, too. I like playing video games and stuff, but I also love basketball, so I'll be playing basketball a lot too.
Oh, well, Chad, you're the next.
Okay. Hi, I'm Chad. I'm from Georgia. I just finished my freshman year. I'm an applied math major. I enjoy music and cooking.
Great. I know Rusty also like to cook, so you three can match and then we can have fun. I have a lot of experimental food for us. One of my biggest interests is to eat. So, so you pick my interest. That's good. Okay, Michael.
So my name's Michael. I'm from Sandy, Utah as well. So woohoo. I'm a music performance major. What? And I also like to cook.
Great.
I have a few friends who go rock climbing, so if you guys want a mutual friend to go rock climbing, yeah, that's about it.
2-year-ish.
Great. Well, I'm really excited that we have like a big groups of students, like you guys joining us this semester. So like really briefly, just introduce our lab a little bit. So Dr. Price is there, and we have two postdoc, Brad and Lena. And like, so I hope this like organization chart is clear enough for you to kind of know what we have there. So Brett is like you're going to learn about their project like later, just really briefly. But we have like programming project, like help a tool to help us do like analyze all our data. That is Brett is on top of it, and Lena is doing protein fractionations, and myself is doing folding, and Rosie or Ken is not here. We'll hear from them later, but they're doing lipid stuff. And while all of the other are mostly focused on protein and like mass spec imaging. And Nate is doing a nice like mouse study on like protein and like dietary restriction and ApoE like Alzheimer project. And Rebecca here is doing like brand microsampling. And so, and underneath are their undergrad. And for those doesn't have picture, is like what we need people for. So at the end of the training, which is like not next Monday, but Monday after next Monday, like we are going to like, you will be revealed which project you're going to be. And then, so, and I hope I did not catch you guys pictures wrong from the Facebook.
Picture
No, you're good. Facebook, right?
Yes. It is like, I was like, okay, I'm half sure that is Michael and Tyler.
No, that's my one picture on Facebook. You got it.
And but because there's like more like Tyler big man than what I expect on Facebook, and then I only know you guys by your eyes, and so I'm not like I get that one. So now we're going to hear from Roger, and before that, like JC will talk to us first.
Okay. Well, we're glad to have y'all. Lavender has done an awesome job in organizing all of the training. It helps to make sure that everyone's on the same page moving forward. And what we're trying to do is just make sure that your time here is efficient and as effective as possible, that you can make a meaningful contribution and be on some papers, right? That's really the goal. If there's anything where you've got questions or concerns, please come and talk to me. This organization is again meant to help you, right? So that you can always know, someone knows about the project that you're working on with the nitty gritty details, but always feel free to come and talk to me if you've got questions or problems. I'm more than happy to help. Especially during this last semester, I didn't really have much time for anything, but during the summer, it's a lot more open. Yeah. So other than that, one of the problems that we run into, so you'll see Lavender put collaborations there. One of the problems we run into is that the skill sets that you're working on and developing are applicable to an amazing number of projects, right? And so the problem we run into is actually having too many things to do and becoming too diffuse. So in the beginning, one of the things that I'll try and do is make sure that you are working only on projects that really feed into the overall goal of this sort of idea. Because if we start getting too broad and too working on too many things, it doesn't lead to conclusions that are publishable, right? And the reason that we want publications is because those are very high value items that you can put on your resumes and your CVs as you go and you're thinking about your next job, your next step in your education. These are things that as I interview students or as people I know that are in professional programs interview students, having a paper really brings you up above a lot of the other interviewees because it shows that you can take a problem that no one knows the answer to. You can ask questions that lead to answers that meet review by other peers, within the scientific community. And so that's going to be our number one goal. Again, if you have any questions or concerns, come and talk to me, okay? Is there anything else that you wanted me to say?
That's good.
Okay, I'm gonna go check on the instruments. And so I'll be here tomorrow. Today is technically a day off for me, but I'll see everybody tomorrow, okay?
Thank you.
Okay.
Hi, JC.
Yes.
So. See what else do you say? Just send me an email. Okay, so Brett will be on next. Why I need to here. Well, his presentation I email you. Yeah, sure. Thank you. Thank you.
You
You're
can just
doing
take
that.
this back. It's
Yeah, you can just take your computer. But let me just take that one. Okay, so as Brett is pulling up his presentation, just like for those who have notebooks right here, just like start, like if you want to take note from page 5, starting from page 5, and then we will go from there. And then feel free to ask questions if you want to. So Brad is like, so just like each of the project lead, or like they will assign their like members to present their projects. So it is a roughly 10-minute presentation for each project to have you like to have a glimpse of like what we are doing in the lab generally. And then we really, at the end of the day, we really want to find something that like really motivates you, that you really like, and then we hope that will be your project. So like we take that into consideration. So ask questions if you want.
okay. So I am the lab coding guy. My name is Brad Naylor. I am a postdoc. No one really works under me directly. I consult on several projects, but that's kind of how I am. So just to get a basic idea of what we do in the lab. The main thrust of the research we do has to do with proteins, right? So in basic biochemistry, you take amino acids, these circles here, you put them on your tRNAs, and then you put them into proteins with the ribosome in an order dictated by your messenger RNA that you got from the nucleus, right? So what we're interested in is how fast this process of how fast these are created and then destroyed, how fast that happens. Now the amino acids are just reused quite a bit, and you get some from your diet, and there's several pathways where they can turn into other things and so on and so forth. So what we do is we introduce a label, this green color here. So what we do is we use a heavy isotope. So we use deuterium, heavy hydrogen. We introduce this into the diet of a cell or an organism or a person. It is when the cell makes amino acids, this extra deuterium is stuck in, and then on some probability, these green dots will make it into the actual proteins, and then we can measure that, and then the number of labeled versus unlabeled is how fast the protein is turning over, how much is turned over at that time. We measure this by mass spectrometry. Then I know we've got a few sophomores, so you may not have actually seen any mass spec data. The mass spec is a very, 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 very precise scale, essentially. That's what JC went to check on just now. And when you get mass spec data, normally for peptides, so small pieces of a protein, it looks like this. This is all one peptide here. This is all one compound. These different peaks are isotope peaks. So this compound has some number of carbons, hydrogens, nitrogens, oxygens, and so on. in this peak, this 50% of them have no extra neutrons. About 35% have one extra neutron somewhere. Now, some of these will have an O17, some will have a C13, some will have a deuterium, and so on. This has 2, this has 3. And this is all just based on pure probability. So what we do is we say, okay, we've provided deuterium. If we have no extra deuterium, the value should be at these blue lines. If all of the protein is new, all of our deuterium is incorporated at the probability we provided it, we should be at these red lines. We're actually at these black lines. And so we just say, okay, how far have we gone? How far do we have yet to go? Do this at several time points. So we do this with this mouse experiment. This is a fun mouse experiment. We're actually gearing up to do one of these. We inject the mice, we give them D2O on their drinking water. We take their tissues when they're at 5% D2O, which gives us a constant deuterium enrichment at 5%. And then with that, that tells us where these red lines should be. Here's the blue lines. And then this particular one, we're about 50% of the way done, which corresponds to this time point here. We do this at multiple times and just fit a basic kinetic curve, 1 minus e to the negative kt. And then the turnover rate is this value k right here. So this is the basic math the lab is kind of built on. My job is to make software that does this. So I maintain our code called Deuterator that does this. My current project is extending this to humans because for humans, you cannot inject them with multiple liters of deuterium. You cannot control their drinking water. You can't get them to 5% D2O, and you can't cut their entire liver out to analyze it. I don't know why, but they get really twitchy when you suggest doing that.
Maybe we can get like tiny pieces.
They actually don't like doing that with liver. The liver takes a long time to heal, so that can lead to bad long-term consequences. They only do that if they suspect a serious liver disease. So what we do is we give them some water and we take their blood instead of their liver. And so the problem we run into here is the water. This is the deuterium enrichment. It's constantly increasing. And so I can't tell, the protein rate will determine how much water is involved. So it turns over really fast. It'll only be using this deuterium here and make these blue lines here. I keep changing the line color on you, I'm sorry. And if it's a very slow turnover, it'll use this entire trapezoid integral here and make these lines. And so we need a recursive function to do that. And so we try like do this equation, do some math hundreds of times. We find a minima and we figure out a perfect fit that tells us the K through math that I don't understand, so you don't have to either. But this is what I am doing currently. But mostly for the purposes of you guys, I'll be interacting if you have questions, and I'll be maintaining the code that does this for your experiments. Does anyone have any questions about any of this?
And he's super helpful if you're coding and having trouble.
Yes.
All the time.
Yes, most projects will have a dedicated coder or a couple people with coding experience, because when dealing with large data sets, coding becomes necessary. There's a handful of people in the lab with different expertise. I'm good with Python programming. Isabella's good with R. JC, the big boss, can do amazing things with Excel. He was doing all of this in Excel. He can make it dance. But anyway, but yeah, we have several people in the lab who are skilled. If you have skills or inclinations that way, let your project leader know. They'll have some way you can help out. Okay, no questions?
So like, if you have your presentation and haven't sent it to me, please send it to me because I'm recording. So we need to make sure all this like presentation is done on one computer. So.
Can I use a flash drive?
Yes, and if you want to use a flash drive, you can do that too. Yes, that's right there. So the next one is from Lena on the amazing of protein fractionation. So, just like, do you guys remember in the interviews, like during the interview, I ask the questions, ask you to ask questions, and just like explore your actually rated on how you handle that questions. So I would like you to start asking questions if you can. It is not, well, now you're going to not, like you will not be evaluated on asking questions, but asking questions is a really important technique. So if you have any questions, don't leave it to the end and just try to ask it. And because there's no dumb questions, I know how you might feel like I don't want to ask the questions to show I'm not prepared, because I was that way in classes, and most of the time I did not read my preview material. So I am worried about asking questions. But here, like, it is not a class setting, so feel free to ask any questions that will help you. So, and that is a good habit to start with. Yeah.
So most of our research is based on proteins. So David and I work on protein analysis. So which proteins we are analyzing is, you can see we are detecting low abundance proteins. There is something called as high abundance proteins and low abundance proteins. So we'll see what these high abundance proteins and low abundance proteins means. So proteomics is a study of set of proteins, and we study this proteome. In this proteome, we have low abundance proteins and high abundance proteins, which make this proteome. But 90% of the proteome, that means 90% of the proteins which are present in our body, they are high abundance proteins. If we see human proteome, it contains more than or approximately 20,000 proteins. And 90% of these proteins are high abundance proteins. So only 10% of the protein, they are low abundance proteins. But this 10% proteins are responsible for the diseases which are present in low abundance. So that is why it is important to detect this low abundance proteins. So this is an example of human blood serum. If you go to the plasma center to donate your plasma, this is what actually you're donating. And if you see, then this entire part of the proteome here is high abundance proteins, and only the pink part of it is low abundance proteins. And to understand the diseases, we really need to understand those proteins. And to understand the proteins, we need to separate, and not only separate, but we also need to identify what those proteins are and how they are making the difference. So we separate that pink part of it that you are seeing on the proteome and then identify it. It is like you have a forest, you separate it into trees, and then you identify each tree, what it is. So what is exactly happening and how we are doing it? Why we are doing it? Because those proteins are responsible for the diseases. How we are doing it? We are using HPLC. It is called as high performance liquid chromatography, earlier known as high pressure liquid chromatography. So you can see a fancy instrument over here. It has 4 parts: pump, injector, column, and detector. On the right hand side, you can see the structure of column where actual separation takes place. What we are doing is we inject protein mixture on it. And if you consider those red and yellow are different proteins, then they interact differently with the beads which are present in the column, and then they get separated. So if we look at the in depth, what it is, so the same instrument, and then we have pump, which pushes the solvent in the instrument. We inject different proteins, like you can consider There are two types of proteins over here, which we have seen, like the purple ones and the red ones. They interact in the column, interact differently. That is why separation occurs. We cannot see them by eyes, so we have a detector on the system which identifies it. It is a UV visible detector. And then we get the separation. Depending on the separation or the fraction which is coming out from the column, we collect the fraction. That is how we are separating proteins. So it is protein fractionation. And then we get a nice peak. Yeah.
So you said there are a lot of low abundance proteins. How do you get clear separation?
Yeah, so that is, I'm going to show you after this. How do we get separation from high abundance to low abundance? So this is the instrument which separates it. And we can see that in the form of absorbance. And that is how we know that this is low abundance and this is high abundance. And that is on the basis of conjugation which is present in the molecule, because we are using UV visible detector, which detects the molecule on the basis of conjugation.
Are we separating individual proteins or just like the groups of high abundance and low abundance from each other?
Just the groups of high abundance and low abundance, because it is difficult to separate each protein. Those are more than 20,000 proteins. So we separate them on the basis of abundance. Now this is a nice chromatogram, if you see, very symmetrical, very nice. But then the actual chromatogram, how does it look like? So it looks like something like this, with a hump and a bump and not a clear separation. And we really don't like this. But then this is what happens when we inject the sample for the first time. And our job is to separate this as much as possible. So what exactly we are looking into it? So if we look at the chromatogram, it is actually, there are many peaks hidden behind that big peak. And our job is to separate these peaks, because we want to separate this peak, and UV visible detector is helping us to see it, because we need to see it if we want to separate it. And the detector is doing its job only when we are optimizing it. We need to optimize when we are working on HPLC. We need to optimize parameters like mobile phase. There is something called as isocratic and gradient mode. Isocratic, where the mobile phase composition is same all the time. Gradient is where you change the mobile phase composition. And for this complex separation, we always need to use gradient mode. And there are different gradient modes that we can use. So we change the parameters, we change the temperature to separate out these low abundance proteins, which are hidden behind that big peak. And then what do? We know this is the absorbance signal. This is absorbance versus time. So we divide them now on the basis of absorbance. You can now see the low abundance peak giving us low absorbance and the high absorbance. So we separate them. We separate the fractions and then we chop off. So we chop off these proteins, low abundance separately and high abundance separately, so that high abundance cannot interfere with low abundance. And after separation, in the mass spec will able to identify it, because now there is no interference from high abundance ones. But then mass spec, we are doing bottom up proteomics. So after separating, after protein fractionation, we are digesting these proteins. That means we are chopping off these proteins into peptides, and then that sample goes on mass spec, where we identify proteins, and on the basis of that, peptides, and on the basis of that, proteins. So the goal is to separate these low abundance proteins from high abundance proteins, because those are biomarkers and diagnostic tools. Concentration of these proteins, when it changes in the body, disease occurs. And that is why it is important to study these proteins. And also it is a key to understanding the diseases. So basically we are doing HPLC separation, where we are just separating, we cannot identify it on HPLC. And that is why we are putting that mixture further on MS, where we can identify what exactly that is. Any questions?
So does your gradient go from like polar to non-polar, or is it non-polar to polar to get that kind of separation?
So it goes from polar to non-polar. Also, gradient depends on different kind of columns we are using. There are different modes of chromatography. In the lab right now, we are using weak anion exchange chromatography and reverse phase chromatography. So in case of reverse phase chromatography, it goes from polar to non-polar. And in case of weak anion exchange chromatography, where the salt gradient is used, it goes from higher to lower. So it depends on the mode of chromatography which we are using. Any questions?
Thank you, Lena. So, like in proteomics, like once later, you'll find it is interesting that we are all in a mode of looking for higher numbers. So, like Lena had introduced, we have 20,000 of proteins in the our genome, in our proteome, and then it is not really a race. But on all the publication, you can see people reporting, hey, we identified 8,000. Okay, that's a number I make up, but yes. So with what what Lena is doing, then we're able to bump up that number so we can understand more about our proteome. So now we'll have Isabella to present on coding. And good job asking questions.
So I'm Isabella, and I'm presenting sort of on Lavender's project. And we've been developing a method to measure the folding stability across the proteome, but we've been focusing on transferrin as sort of a model system. So we're particularly interested in measuring protein folding because if you remember that diagram that Brad showed at the beginning of his presentation, where if you're looking at sort of a protein's life cycle, they get synthesized and they get degraded. But what we haven't been able to measure yet is how well they fold. And protein folding is a really important measure of protein quality because the folded structure of a protein determines its function. And as you can see in this diagram, if the protein becomes too stable, then you can get aggregates or amyloids, and those lead to diseases like Alzheimer's disease or TTR amyloidosis. And if they are not fully folded, then that would be at a higher energy. So a protein has a correct folded state with a specific stability that allows it to function correctly. And transferrin is sort of the model protein we've been using for this. It's in blood serum. It transports iron from the blood to the tissues. It has 2 iron binding sites. There's sort of one on each side of it. And the binding sites are not exactly the same, but previous research has shown them to be stabilized when they bind to iron and destabilized when they release iron. So when they're not bound to iron, they're flopping around more. And we hope that we can measure that both in purified protein and in the complex mixture of blood serum. So this is how the folding assay works that Lavender has been developing. So we take the sample of proteins and we split it into 9 fractions and then add an increasing amount of guanidine to each fraction. And guanidine is a chemical that disrupts the folded structure of the protein. So that allows amino acids that used to be on the inside to, when it's unfolded, they're more on the outside and exposed to the solvent. And then we add iodine to label some of those exposed amino acids. And then you can see here that when we fully unfold the proteins, as you added more guanidine, there's more iodine on the proteins. And then we do standard mass spec filter prep, run the samples on the mass spec and analyze the data, which is quite challenging sometimes. So this is the kind of data that we get. We have a list of different transferrin peptides that we get, and this is not all of them, we can get hundreds. And for each of those peptides, we can calculate a C1 half. So that's what this graph is. It's like the amount of the protein with the iodine modification as a function of how much guanidine we added. So you can see here that as we add more guanidine, there comes a concentration where the protein starts to unfold, and now suddenly all of it is iodinated. Does that graph make sense to you guys? And based on how much guanidine it takes to get you to that point, that's a measure of how stable the protein is. Because if it took much less guanidine to get to that point, then that would mean that the protein is very unstable and you barely add any and it will.
I have a question.
Yeah.
You're working with transferrin because you have this like control idea of how stable it should be?
Yeah.
Okay.
And we know that we can like add iron to it and it should become more stable. It's just very well studied.
Okay.
Yeah. And by the way, the Y plus 125.9, this is iodine here. And this is 2 iodines. So then we can take all of the peptides that have the same labeled amino acid and then take the C1 halves that we measured for each of those proteins in different conditions and plot them to show how the C1 half changes under different conditions. And here apotransferrin is transferrin that is not bound to any iron, and holotransferrin has iron in both binding sites. So we can see that for this particular labeled amino acid, it gets stabilized when iron is bound. So the main hypotheses that we have been testing are that our method can measure changes in the folding stability of transferrin, and that we'll see an increase in stability, especially at peptides around the 2 binding sites when iron is bound. So the 2 experiments that we did are loading iron onto apotransferrin to see if the stability increases, and using EDTA to remove iron from holotransferrin to see if the stability decreases. So this is just how the iron removal experiment works. We have just plain apo and holotransferrin as controls, and then we incubate the holotransferrin with either water or EDTA and centrifuge it to remove any cations or EDTA, and then do the folding assay on the protein that's left on top of the filter. So the first thing we did just to check that this was working was UV-Viz, and Dr. Watt showed me how to do this. You can add bipyridine to a sample and it makes this lovely hot pink solution if there's iron, and you can measure it on the UV-Viz and get this peak at 522 nanometers. And when we just added water, we detected iron on top of the filter with the protein and no iron on the filter. And we also And when we added EDTA, we detected iron that went through the filter with the EDTA. And that was what we expect to see. So then we were just going to see if the mass spec folding assay results agree with this, because they should. And we found that they do. So this data, we just plotted the C1 half, like every C1 half that we measured as a function of the label site on the protein. Transferrin has 600 something amino acids, and the first iron binding domain is somewhere around here, and the second domain is somewhere around here. And we can see that for the first iron binding domain, holotransferrin has a pretty high C1 half, but apotransferrin has a lower C1 half, and it sort of dips down right here, showing that this domain gets destabilized when there's no iron bound. And when we add EDTA, we see that all these C1 halves that we measure are down here with the apotransferrin. Yeah.
What was the C1 half? Okay.
That's like on that curve I showed you, the point where it jumps up. That's the measure of how stable the protein is.
C1 half is, that's like concentration where 1/2 is something, right?
Yeah.
Okay. Yeah.
So here, I think I was trying to make figures in this PowerPoint and then messed up a bunch of my slides. I should have checked that before. Yeah, so these are some of the results for 2 amino acids that are sort of next to the binding site. Like for this amino acid, we see that the control and holotransferrin have a higher C1 half, and apotransferrin and EDTA have lower C1 half. And it's the same for this amino acid that's also right by the binding site. So that's encouraging, and it agrees with the UV vid results. Yeah, I was making more figures here. So, yeah.
Is there a situation where only one iron bonds instead of both?
So transferrin in the blood is kind of like in a dynamic equilibrium with iron. Like there might not be quite enough iron to bind in both sites. And it's not really clear if one site fills first or if there's transferrin, or if in the blood there's a population of like apotransferrin, holotransferrin, and then transferrin with each of the different sites bound. So I'm not sure if we can tell the difference between them from blood serum data.
That is one of the study we're trying to do.
Yeah.
I want to know if like there's like bias of each binding site. But so far what we see is whenever we low iron, we jump up. So like like it become like a hollow version, which like 2 sides are open. So
but we've also been adding like 4 equivalents of iron per equivalent of protein. So it's probably just holotransferrin the way we're doing it. So yeah, this is our iron loading experiment. We loaded all these different equivalents of iron and then did the folding assay. And this is the data that we got for all these sites that are near the binding site. And this one actually is one of the amino acids that directly binds the iron, which is super cool. We can see that the C1 half increases as you add an increasing amount of iron for all 3 of these sites, which is really encouraging. And then we tried it in blood serum, and this is kind of old data, and we have better data now. I just haven't put it in the slide. Wait, I thought we did. I will check.
the RB one, right?
Yeah.
You're saying like, so it does like have a train.
Yeah.
Sorry, I'm starting discussing my data, so I will talk to you later.
Okay. What I mean is that we just got better coverage.
That's
across
great.
the
If it is like, I was like so nervous. So let's talk about that later. I'm sorry for the diversion, then, but.
We just like retried the EDTA experiment and barely got any proteins, but now we've done it again and like got more different peptides. But we did measure some of the similar things where for these sites near the binding site, if you add EDTA, then the C1/2 goes down. And we've been working on getting better data in blood serum. So in the future, since folding stability is just so important to lots of different things, well, first of all, we want to publish a paper on our preliminary results and then possibly apply this to either Alzheimer's disease or TTR amyloidosis or something else. So, yeah. Do you have any more questions? Yeah.
So with the folding of proteins, like we don't know exactly what it would relate to in the end, like when it comes to like diseases, but we're doing it kind of a larger spectrum, just on proteomes.
Yeah, so we have some more data that I haven't put in this PowerPoint where we just added EDTA to blood serum and we wanted to see how it changes the folding stability of lots of different things in the blood serum. And we did see changes in a lot of different proteins. So we expect that for a disease versus a healthy subject, we would see changes in protein folding stability. And hopefully we would be able to learn something from that, like if certain proteins aren't getting folded correctly in a disease.
Carrying that on to diagnosis.
Diagnosis is one thing, like for like TTR, that's the goal we're trying to do. If like, yes, off table, I don't want to talk too much, but like, yes, that is like one of the possibilities we want trying to use while we're working on serum, is because then we can use stability to try to find mechanisms and then diagnosis. Yes. Single person.
Does amyloid mean resembles amylose? Like, is that why it's called that?
I don't know.
Okay.
Those like, so they're the protein aggregate, and then like they like TTR aggregate together, and that disrupt the functions of the heart. That is like for cardiac amyloidosis that we are working on. Good job asking questions, and then like again, just like research is about curiosity that really drive you, and then so and also like Isabella's slide says, like after we process through the software, where is the really fun digging is when you start digging through the Excel sheet that has like at least 26 columns times like 200,000 of rows. And how are you going to find an answer from that? It really, you need to have a questions. And what questions drive you more is like the questions that you are curious about. So like asking questions is really an important technique in research, and it's just like not asking questions in front of like to others, and also asking questions to yourself, like what I want to learn, what I'm interested in, what I'm curious about. So just like, and then again, I will really force you to ask questions if you don't, kind of ish, like later on you will find. And then, but really like, just try to crack out of your shell a little bit. I know it is a little bit hard, like when we just first met here, but I guarantee you I don't bite and nobody here bite. Okay, so.
Lavender.
Huh?
Speak for yourself, Lavender.
Yeah.
Tyler bites.
Sorry.
Okay, well, don't ask questions to him then. Well, all right, it's okay. I will protect you from him not getting bite. But anyway, so, Rusty, do you want me to download your slides?
Yeah, they're in your.
Okay, yeah, let me just.
You want me to go now?
I'm going to like you're the next, so let me sign in. Email, did you email? Let me just make sure I log into the right email. And then just like, we are the best lab because everybody here have fun. And so, really.
I thought we were the best because we were so damn organized.
Wait, no. No, Oh, please don't do that to me right now. Okay.
Everyone look away.
Oh.
Is it that you're using your Gmail and stuff?
Oh, yes, thank you. Okay, yeah, let me, okay. So, again, troubleshooting is really important. You need to know who you ask your questions to. Can the BYU, the EDU, and there's also, you need to have really talented students, like people working with you, like I always do, and actually, really, do I need to do that? What do I see? 17. Okay. Let me download Rusty's slide first. Wait. No, don't do that to me. Oh, okay.
You said you're a senior?
Apparently, I log into the
Far along. What class did you take?
He's just going to give his presentation.
Oh, gosh. Wait, now you know how unreliable I am. Not true. Okay, this should be.
More interesting or less memorization?
And yeah, as I'm struggling with computer. Well, just I feel better because there's one time JC literally spent 20 minutes in your meeting trying to figure things out. So.
Just
oh my God.
I'm studying for
Okay.
the summer, so I have to
It is download. Okay, Rusty. I'm sorry, but
All right.
the slides are up.
Maybe adopted something.
Okay, so if you guys remember what Brad was talking about with deuterium labeling, is it this guy or this guy? Remember what he was talking about with proteins being labeled with deuterium, where we then calculate rates? I'm applying a lot of those ideas to lipid analysis, and he showed you did you show rate curves?
Theoretical one.
Okay. So we're producing rate curves from these lipids, and you can see that sometimes, like these green ones, this is a specific type of the molecule. It's kind of flat, starts high and ends high. That's not good. This red one, though, looks like a nice exponential curve, and that's really good. And that's good. And then this is kind of that one broken up into two different ones. So that's kind of what we're trying to do is at the end of the day is we want to generate rates from lipids. Okay.
Why is that good, Rusty? Why do you not want to see it coming down? Why do you want to see it coming up like that?
Coming down, it's noisy and more flat than coming down. But we expect it at day 0 when we're not labeled. So each data point, I'm not going to go into the details. That's not my purpose here. Let's go into that though. You're asking me a question, I'll answer it. Okay, at time 0, we don't have any deuterium in the molecules. And each data point on here is the result of the math that Brad showed, and that's called fraction nu. So that's the amount of new molecule that has a label. So if we don't have any label, then what is that fraction? It's always going to be 0. So you can see how these just don't make sense, simply because they might be really noisy or for other reasons. Okay. And then because we're labeling with time, the more time goes, the more of the molecules pool we expect to have the heavy label in there. Therefore, we expect to have the highest fraction nus to a point when the curve eventually plateaus, where we no longer observe more changes. and the theoretical maximum would be 100%. Okay. So he showed you that little schematic from basically the central dogma. You guys know the central dogma? That's DNA to protein. So whenever that happens, the tRNAs are slapping in labeled amino acids into the peptides. Well, we have lipids that are being made, and if you have a precursor lipid that makes another lipid, or serves as a precursor to make another lipid, then we can get the deuteriums added into parts of the molecule that are newly built in. So from fatty acid synthase, for example. Just like each amino acid can get labeled, like Brad mentioned. And so what our workflow looks like, as mentioned, is we take mice, we collect tissue, and in this case, we're looking at Alzheimer's disease models. And so we take half of their brain, and each half should be a mirror image. That's our kind of how we work, is we just make that assumption. So we grab one hemisphere, and we homogenize it, so it turn it into a goo, so everything's mixed together. In other words, we can no longer differentiate the cerebellum from the hippocampus or from the brainstem or whatever else is in the brain. It's all mixed homogeneously. That's why it's called a homogenate. And then we perform an extraction, a chemical extraction, where we get hydrophobic stuff like lipids separated from really, really hydrophilic stuff like carbohydrates or proteins. And then, as Brad showed, we get a difference in the spectra because of the heavy label. We calculate fraction nu, and then we plot those rate curves that I just barely showed you. So this is what we do for liquid chromatography mass spectrometry, is we do this homogenate. Well, what if we were interested in actually looking at the specific parts of the brain and not just turning into a giant goo here? So this, we can't differentiate parts of the brain. What if we did care about the different parts of the brain? Then we do this workflow. We do what's called DESI. Same thing, we take the mouse, we take a brain and we slice it crazy thin. And then we take that crazy thin slice, and this is the mass spectrometer, and we raster across there with a spray. We won't go into the details there, but needless to say, we can map each point that we put in the mass spec using a software to make these images. And we can look at the rates for all the points along here. So this red stuff would be like really high turnover, and the blue stuff here would be like really low turnover. And that's essentially it, right? So what I'm currently doing with the liquid chromatography data, just so that you have an idea, if you look on the board back there, you'll see 4 different peaks. All 4 of those peaks are for the same lipid. That's a problem. So we're trying to figure out algorithmically how to pick the right peak each time. So that's something that I may need help with in terms of the project. And also the other part of the project that I need help with, all your horses can. The other part of the project that I might need help with is this part where we're actually doing extractions and mass spec prep. So doing the mass spec analysis and then doing the mass spec prep on the LC side. Ken is actually the undergraduate that's responsible for this part of the workflow, and that's what he's going to talk about right now.
I can find them out.
You want to talk about this at all, Rusty? You can see it. If we get questions, we'll.
Sweet. Okay, so I'm working on DESI, which a lot of people have talked about proteins. Rusty and I are looking at lipids. So lipids are cooler than proteins. Right here. Yeah, just assertion. So these are what lipids look like. As you can see, they're colorful and really cool. So like they store your energy, they signal. They're like your brain is basically a giant blob of lipids. So cooler. As Rusty said, what we do is for DESI, we want to know how the hippocampus differs from the cerebellum, from the pons, and every part of the brain. And so we actually cut the brain up into tiny little slices so we can map exactly where we are in the brain. And that's kind of the most important part, is knowing where in the brain am I, so that I can tell how the lipids are acting inside that part of the brain. And so we have this little contraption down in, yeah.
You factor in pathways, or does that not really apply? Like where different signals travel through the brain? Like do you section it off into parts?
Yeah, so we're not super that quite advanced right now. What we're trying to do first off is just to get a glimpse of how the lipids behave in the brain. And then we'll look into, okay, now what does that mean? What are the pathways? Like, could it be that these areas of the brain that are focused more on memory have different turnover and concentration of certain lipids? And is that related to Alzheimer's disease? So that's kind of the end goal of the study. So down in the basement, we have a little contraption that does this. It sprays electric solvent. So it's just methanol with an electric charge onto our brain. And all of our lipids will fly up as charged particles into the mass spectrometer, so we can then measure them and learn about them. So what we're actually going to be shifting towards is MALDESI. So what that's going to do is, instead of having a charge put onto our lipids because of just pure electricity, we have a laser that's gonna like make a little explosion on the surface of our brain, charge everything, and we're gonna throw it into the mass spec. And so then you can see here, this is kind of like an example of a brain image. This is what the brain would look like after it's stained. So you can see the different brain regions. And then you can see in here, some lipids are more prevalent in different regions of the brain. Some are just kind of ubiquitous. Some are in the ventricles. Some are more in the cerebellum. And so we can use that to make correlations to Alzheimer's disease. That's the project I'm working on. As Rusty said, we can also calculate the turnover based on the Deuterium Incorporation inside these lipids. And there's a picture of a wave proving once again that lipids are cooler than proteins.
Because water waves mix so well with that.
Well, if you weren't a giant blob of lipid, then you couldn't float on the water. And you couldn't stick to your board because the board is what it's coated in, Ken.
It's coated in wax.
With wax.
It's a lipid.
There you go.
So you're really trying to against all the other projects right here, like prove lipid is more important?
So any
project, just put the other ones down.
You guys have any questions? We're trying to keep it brief so we can interact a little bit more. So you can ask any questions you want.
Or like questions not on the project is fine too.
Yeah, go ahead, Mike.
You get Alzheimer's mice.
Well, they're not Alzheimer's mice. They're Alzheimer's model mice.
Okay.
So they don't necessarily have the disease, but they have a human lipid carrying protein called apolipoprotein E. And we put the human version of that in these mice. And there's 3 different versions of that. One, we've observed just empirically that one of those isoforms is more common in people who don't get Alzheimer's. One's more common in people who do get Alzheimer's disease. And then the other one is considered to be neutral. or the one that Brad mentioned briefly, that one actually is going to be a better mouse model. And those are going to be CRISPR knock-in mice. They're not going to be vector knock-in mice.
Lipid is really cool, and one thing is that really honestly, lipid is harder to study than proteins, because for protein we have all our protein, like maybe not all protein in the proteome sequence, but most of them are sequence, and then based on that we can do the protein ID a lot more accurate than lipid. So lipid group, like they are amazing. Like still think protein is cooler, but like they do the hard work.
Is the project that they're doing the only one that has to do with looking at Alzheimer's and everyone else is focused on the protein function?
Well, actually, like so. Sorry.
What was the question?
I was wondering if you two were working on one that was based solely on lipid functions in Alzheimer's, and everyone else was focusing on proteins and trying to like figure out how they correlate, or like.
Yeah. So, are you doing the protein part of the Alzheimer's project? So Nate's doing the Alzheimer's but protein version. So he's doing the same thing we're doing, like taking the brain and then looking at stuff. And then Becca is also doing the DESI part that we're doing, where we're doing the imaging. We're doing the imaging. She's doing that with proteins.
That's so cool.
So we're doing
Hopefully my presentation shows
that it's being separated into two people, and they're doing the protein versions.
I think I'm the only one not doing anything with Alzheimer's right now. I feel left out right now.
Yours is related. Alzheimer's is primarily a.
So
the
we
main
are like
symptoms are fully
one step before we can get to Alzheimer's is. So anyway, I will get into the club soon. I hope. But yeah, but that is Tyler and Michael that they're going to try to apply what Isabella has been doing onto Alzheimer's disease or something else. So we will see. Cool. Okay. So Nate is next.
So I was about yay high with a head full of hair when I joined this lab.
A warning to everyone who's joining.
Yes. So where I'm
This
going with
and
this
get good data.
is that I joined this lab, and I remember the first day I came. My first day was at a group meeting, and JC was up there, and he was just blasting away, talking about mass spec and isotope peaks and M over Z's. And so, if you feel lost, that's okay. But your hair will be gone by the time you're done.
We have the razor back in the lab. That's what we're doing. That's what we're doing at 2:00 today.
You see, I'm not very inspiring today, but I promise that something I will say is hopefully going to be helpful for you.
If the instrument's working, then that's very inspiring.
I mean, you're not wrong.
Hey, I'm finally off my other TA.
Yay!
Ready.
Officially.
Like,
Well, actually, yeah, you're the now. So Rebecca is inspiring, and Nate is
he's helping.
and Nate is watching the whole thing self-destruct on the 6560.
Like he's now the overall like director of everything out there.
Okay, so fellas, if you have any questions, please stop me and ask. So don't feel scared. I'm not going to talk a lot about the details and the mass spec stuff. We can learn that on the way, because everything I say you'll forget. But today I want to talk a little bit about what we're doing, and the focus, our primary focus right now is also about ApoE. We're studying not just proteins, but we're studying the proteins that are involved in the lipid metabolism. So we're kind of like a little bridge in between those two projects. So just want to start really quick talking about what apolipoproteins are. So a lot of what happens in your body when it comes to lipid transport happens through these little vesicles called chylomicrons. So these are just like little capsules that carry lipids throughout your body. And one of the things that apolipoproteins do is that they bind to the surface of these lipid vesicles, and they kind of help with knowing where to dock, binding to certain receptors. And you kind of heard about this. There's LDL and HDL. Both of those are basically a lipid vesicle that looks like that. LDL means you don't have a lot of proteins in that, and HDL means that you have a lot of proteins on that thing. That's just a very simple thing. And ApoE is one of those little apolipoproteins that we are going to talk about. So ApoE is one of those little red things. And ApoE is made by liver cells, and it helps deliver, and it's involved in delivering cholesterol to the brain and to other tissues to make things work. In the brain, ApoE is made by astrocytes, and it also helps deliver cholesterol to neurons. It's also involved in immune function to help macrophages work right up in the brain. And we've talked a lot about ApoE and its variants. And a lot about what we do isn't necessarily to do with ApoE itself, because that thing has been studied the crap out of. We know that there are 3 ApoE variants. There's ApoE2, ApoE3, and ApoE4. ApoE2 refers some kind of resistance to Alzheimer's disease. But it does come with a higher risk of other things. So we'll talk, that's not important for right now. ApoE3, you're good unless you don't take care of your body. You're still screwed if you don't. Your body's a temple, so take care of it. ApoE4, now this is our main guy of interest because it is highly involved with Alzheimer's disease. It's also involved with other kind of cardiovascular things that are happening up in the brain. And it's also correlated with lower size of hippocampal volume and all that kind of shenanigans. So that's why we're interested in it. And so again, why ApoE? Because of all of these things that we see are involved with the ApoE gene itself, whether you have 2, 3, or 4. And so our hypothesis is that ApoE4 negatively impacts the way lipid metabolism proteins impacts the lipid metabolism by changing how lipid metabolizing proteins bound to membranes influence all of this.
Nate?
Yes, sir.
So the tubes that you were talking about in the first slide, I can't remember what they were called, apocol something.
Microns, the capsules.
Yeah, where are they just all throughout your body? Or are they specifically in your brain?
So they're everywhere. So when you go get a cholesterol test, that's basically what they're looking at.
Okay.
And I'm not exactly sure how the test works, but yes.
So does each person just have one type of these ApoEs? Is that just a genetic?
Well, you can be heterozygous.
Yeah, so you can have a mix of both. And there's actually, I don't want to get too much into the details, but there's actually different kinds of Alzheimer's disease depending on what variation of the gene you have and also what combination of the ApoE genes you have.
Familial and sporadic are the 2 main ones. And the ones, when we talk about Alzheimer's disease risk with ApoE, we're talking about sporadic, not familial.
Well, it's more of like the Alzheimer's disease itself, not whether it's genetic or not, just the way it influences your brain and it causes dementia and all that kind of stuff. Someone had a question.
Does good or bad cholesterol affect that at all? Like just kind of the interactions with the proteins, and are you looking in that way at all?
So if I remember correctly from basic biochem, the thing about HDL and LDL isn't necessarily a relationship between good and bad. It's just kind of what they do. LDL is involved, it's basically a capsule that is just full of fat. And so you technically, that is not very good. And that LDL is basically going throughout your body and delivering more fat. HDL is kind of like an empty vesicle that just picks up the fat and takes it back to the liver for metabolizing. So in that regard, if the process of delivering cholesterol is not very good, then your other cellular processes aren't functioning appropriately. And the thing is about cholesterol is that, again, it's neither good or bad. It's involved in making hormones. It's involved in the fluidity of your cell membranes. It's involved in, what's it called? Immune processes in the brain. So if those things aren't working right, then that's pretty bad news. Did I answer your question? Yeah, perfect. Okay. So like Rusty was saying, we have the Mighty Mouse that we use as an ApoE Alzheimer's model. And so we have these three different genotypes that have been, these three different genes that have been introduced into these mice. And what we're doing is we're using these mice to study Alzheimer's disease. Well, not, I take that back. We're studying these mice to see what the influence is of Alzheimer's, of the ApoE gene.
I'm sorry. You have plenty.
I'm so offended. Just to see what the impact is of having certain ApoE gene variants and how it impacts lipid metabolism. So a little bit about probably Brad, if I remember, if I heard correctly, he's already kind of talked about Deuterium Incorporation. We're also going to get kinetic curves from each of the mice, but I don't want to talk too much about that. Again, basic overview, we take our mice. Who is vaccinated? Who here is not vaccinated fully? Back there?
I get my second Thursday.
I just want to take my mask off. Sorry, no, that's a side story. Just okay. So
But we can take out like 51.
no, it's all good. Last thing we want is OSHA here to tell us how we're violating everything. Anyways, so we have our mice models. We take their brains, we homogenize them. And now normally what you're going to see is that you get two little pieces of important tissue. You're going to get your cytosol, the liquid, which has all the liquid proteins that are bound to liquid stuff, shenanigans. And then you're going to get a little pellet. And that little pellet is a bunch of fat, a bunch of cell membranes, a bunch of DNA, and a bunch of proteins that are bound to those membranes. And so those are the proteins that we're interested in studying because they are heavily involved in lipid metabolism. Once again, this is just a very simple thing. This is your membrane. You have a bunch of proteins that are bound to that membrane. Those are extremely important because they are heavily involved in signaling. They're involved in lipid metabolism. And they're kind of a thing that we kind of lay to the side because it's kind of difficult to work with because lipids are messy, as these two gentlemen can attest. And so that's what we're primarily focusing on. And just, did someone talk about method prep and stuff?
No, not
Okay,
yet.
then we're not going to worry about it because you'll see it. But basically, we just take our tissue, we make a smoothie out of it, we separate it into two little pieces, and we want to focus on the non-soluble portion, which is basically this little thing at the bottom. Okay. And so a lot of what we've been doing this last semester has been troubleshooting. We finally have a method that works both with preparing these proteins for mass spec analysis and the best method to analyze them with mass spec analysis. So what you'll be doing is not just working with membranes, not just filter prepping, but you'll also be more involved with the data analysis. And that's kind of one thing that I've wanted the undergrads that are involved with me is to be more involved in the data analysis so that they can understand what mass spec is and how to look at the data and not freak out when they come to JC's meetings and they hear him talk about all these crazy things, but actually understand what's going on.
Who's the lady?
Good question. I think she's from a soap opera.
Why is she on your slide?
It's a meme, Rusty. Also, this is the eternal dialogue between me and Rusty. Me's trying to share a meme with him and him like, I just don't understand this. That's because he has better things to do with his time than look at me, right? Okay, and this is the team that I'm working with right now. There's Kim Lavender, as always. She was my grad student, so I sometimes still steal reagents from her drawer. And she knows that, right? I'm just kidding.
It is not sterile, I was sure with
yeah.
whoever in my group, right?
Yes. And then there's Katie, there's Ethan, our lovely gentleman right there, and Noah. Those two ask a lot of questions. So it's really good to work with them. And then Lena has also been working with us with developing LC methods and stuff. Questions, concerns, doubts, uncertainties.
Always save the best at the last. So Rebecca,
I only get 5 minutes. I can only get 5 minutes.
well, no, like you like so. No, you go whatever, however long you want, and we'll take a break after that. And then they will stop with me
And
wherever.
then after the break, only undergrads will present for 20 minutes. Of this.
30%.
You guys ain't getting out of here till 5.
Okay, so we're working with Alzheimer's disease and microsampling in proteins, kind of like the DESI, but with proteins, as Rusty said earlier. So background, we've talked a lot about Alzheimer's disease. But specifically for us, we're looking in 3 regions of the brain. We're looking at the hippocampus, the visual area, and the entorhinal to see which ones are like specifically affected, like the hippocampus and the entorhinal, that deal with the memory that seriously gets affected by Alzheimer's disease. And then the visual is more of like our control for us looking at how the proteins are affected within the brain. We've done a control where we've had ApoE inserted into mice, but not in the place where it actually was regulated. So it's more of like, it doesn't have much effect with Alzheimer's disease. And then we're getting mice that has the human ApoE knocked in the right place to regulate it. So we're hoping to see changes between the 2. These are pictures that I put in. So you have a healthy brain, and then you have, I guess, an advanced Alzheimer's brain. And we're seeing that you'll go from having like built up amyloid plaques and how proteins also affect everything. So the gap in knowledge, the connection between the ApoE alleles and Alzheimer's, specifically in the regions of the brain that we're focusing on. So we've got the hippocampus right here. We've got the visual area right there. And then we've got the entorhinal cortex there. This brain is flipped. Generally I have it the other way, but whoops. So goals, we're going to try and analyze and understand the regulation of the proteomes present in the 3 regions, trying to be able to reproduce the proteins within the region over like time course brains, measure the concentration, kinetics for how fast they're turning over, things like that. Workflow, we'll take a labeled brain over the time course. We cryosection, which cuts it into very, very thin slices, and they look like this prior. I'm just going to point everyone basically in here. Then for microsampling, we take small little squares like 200 by 200 microns and from each of the regions, so we can try and map out how far out we go, if the proteins change or if they're very similar, so we can be reproducible. So we're going to try and map that. We get them into a well plate because they're very, very tiny, digest them, run them on mass spec, and get data. One thing we're trying now again is we're trying to take the whole regions, like whole visual or entorhinal or the hippocampus, to just see what's in there to begin with. So this is one of the ones that we worked with in the past. You can't read those numbers, but that's okay. That's another brain. Brains get distorted kind of, so we have to really work on our mapping, which is one of the things that we are working on this summer, is being able to map, are we in the right region? Is this really the visual? Is this really the entorhinal? Or anything like that. Conclusions. We've been able to reproduce about 3,000 proteins. We've started to kind of see some patterns, but we want to see if the proteomes differ between the wild type that we've already done and the knock-in mice that we're going to be doing this summer. And then do the proteins vary from the very center of the hippocampus all the way to the edge? Because if it does, we'll need to map exactly where we're pulling for the data. And that's all I've got so far. I'm still working. Questions on very short presentation, Michael?
Oh, no, sorry, I was about to clap.
Okay, good. I'll take clapping questions too.
So you're trying to find out if the ApoE3 or E4, whatever ApoE you're looking at, is building up, or if it's got a high concentration in those areas. What specifically are you looking for about that ApoE, if that makes sense?
I'm still learning what's happening in my own project. But from my understanding is we just want to see what proteins are there between the ones that are heavily affected by Alzheimer's disease, which are the memory, which is the entorhinal and the hippocampus, and see, are there different proteins in there versus the visual? And then are there different concentrations within those regions as well to see how Alzheimer's is affected and stuff like that? That's my understanding.
We don't have Alzheimer's mice. We have Alzheimer's model
mice.
So they don't have symptoms or anything.
APOE acts as like a transcription factor and does a lot of cell signaling, so that's why the proteins could be changed as well.
Yep. But proteins control lipids. So should be able to connect with the proteins at some point and say, hey, science!
Yep.
Then you can get a paper and never think about it again.
We'll ask you about the paper.
You will think about it in your next. Okay.
Everything else.
So, I hope we don't want. Okay, so I hope you guys kind of see like, even though each group have different focus, but if you're thinking in maybe a giant paper considerations, we're discussing like the theme is Alzheimer, and we talk about lipid, we talk about protein, we look at the brain, and we also put into like the possibility of all those proteins that associate with Alzheimer. So each part are all really important, and so it all contribute to the same goal, like the same thing. And as we say, like in our lab, really what we're trying to study is like use a systematic view to look at lipidomic and proteomic. And then like, because like really, one thing I really always being proud of being in our lab and are always really excited is a lot of other biochemistry lab, they look at couple proteins. Like even there's another lab in this building that they're also doing Alzheimer's disease, but they look at just like couple of the protein that they are really solely focused on, like tau protein, like, and then but what we're trying to do is we're trying to look at like what else are affected, because if you have done your biochemistry classes, if not, I just warn you, like the signaling pathway is a monster, but signaling pathway is important. And it is just like, there's so many interactions between all the different pathway, like knowing one pathway is not enough for you to solve the disease or like to prevent it. So that is what we're doing, and that's why we're cool. Okay, so any remaining questions? We're going to take it after we did take a break for 10 minutes, and then you will go into talk with me for the rest of the day. Yeah, so thank you guys. So like Untitled Chapter